everyone, Group 5 will present to all of you about the role of RB protein in cell cycle, specifically at G1 checkpoint. Before I explain about retinoblastoma protein, let me introduce to you to a transcription factor named E2F. So E2F plays an important role during G1 phase as it will bind to a DNA sequence to produce protein for a DNA replication during S phase afterward. Next, RB protein is a tumor suppressor, meaning that it will prevent the formation of tumor, or we can say that it acts as a brake in cell cycle, like how brake works when a car is moving. How does it work? So, active RB protein will bind to the transcription factor E2F as a complex, and then this complex will then bind to the promoter of the DNA sequence to prevent transcription from occur. For the next part, my fellow groupmates will further explain about these three informations that are related to RB protein. Thank you.
Now, Hui and Vimal will discuss about the role of RB protein in apoptosis using the online forum.
Hi Vimal. In a previous lecture with Dr. Jam, we have learned about RB protein. So, do you know what is the other role of RB protein other than in cell cycle regulation?
Hi. Okay, let's continue. The retinoblastoma gene and its protein product, which is RB, have been studied intensively for their role in development of oncogenesis, cell growth, and also cell differentiation. Do you know that RB protein is also involved in the previous topic that we have studied before?
Oh really? Which topic is it related to?
Apoptosis, yes. RB protein plays a role in apoptosis.
Oh, that sounds interesting. Can you briefly explain to me about the mechanism?
Yes, sure. Here we go. Okay, in normal cells, during the G1 phase, the active hypophosphorylated form of RB1 binds to the E2F transcription factors. Now, E2F is inhibited due to the binding of RB. This represses the expression of both cell cycle and also apoptotic genes. Meanwhile, in the S phase, RB1 is inactivated by the presence of mitogens. This will release E2F factors to induce transcription of the cell cycle genes. A fraction of RB1 and E2F complex remain at the promoters of the apoptotic genes. Thus, this will prevent their expression.
Oh, actually I've read some articles saying that in cancer cells, RB protein can be inhibited by either mutation or hyperphosphorylation of RB protein. The inactivation of RB protein will then lead to the activation of cell cycle and apoptotic genes. However, hyperphosphorylation of RB protein can only affect genes of cell cycles only. This actually gives an advantage to cancer cells to proliferate and survive because the cells are protected from apoptosis. So, in cells lacking RB protein, tumor regenesis can occur only if survival pathway protects cells from RB protein loss-induced apoptosis by limiting E2F pro-apoptotic activity and also p53 pro-apoptotic pathway. So that's all from me.
Oh, that sounds very interesting. Thanks for sharing.
Okay, so I hope to see you soon. Bye.
Bye.
So now Ameera and Man Fei, also Nafiessa, will present about retinoblastoma, the eye cancer in drama way.
Nafiessa, you may enter now.
Hi and good morning, doctor. May I know what is happening to me?
Okay, from the MRI and the CT and all the symptoms that you have, I can diagnose that you have a cancer called retinoblastoma.
What is this cancer? Why my daughter will get this kind of cancer?
Okay, you calm down first. First, the retinoblastoma occur when nerve cell in the retina develop gene mutation. This mutation can be caused by lack of RB protein or defect of RB protein in the cell. This mutation causes the cell to continue growing and multiplying when healthy cell would die. This accumulating mass of cell form a tumor and the retinoblastoma cell can invade further into the eyes and the nearby structure and can also spread to other areas of the body, including the brain and the spine. Actually, this kind of cancer is very rare occur in an adult.
Doctor, can you tell me what is RB protein? Why does this RB protein sound so important?
Well, the full name of RB protein is called retinoblastoma protein. Actually, the RB protein is a tumor suppressor protein that is dysfunctional in several major cancers. RB protein bind to particular transcription regulator and prevent them from turning on the gene required for cell proliferation. Transcription is a process in a cell division, and transcription regulator is a protein that is involved in the cell division. In conclusion, the function of RB protein is to control the cell division. Am I make myself clear?
Yeah, we understood.
Okay, nice. So now, Nafisa, you need to be hospitalized as fast as possible in order to control the tumor and prevent it from spreading. I will brief the details of the treatment after you are hospitalized. And so now you can go to check in for hospitalization.
Okay, thank you, doctor.
Okay, welcome.
Hi.
Now, Winnie and Firdaus are going to present you about Mitogens stimulate cell division through retinoblastoma protein with a song that we created.
Hey, Firdaus, you know about cell cycle?
Yeah, sure, I know.
But I'm a little confused, like, what does Mitogen do with it and what's RB?
I got you. Let's listen to this song. Oh, mitogen, responsible for cell division. When it's binding, cell proliferates from the G1 to the S stage.
But RB has a control negative. Without mitogen binding, stops cells from proliferating. RB holds transcription regulators in inactive state and disturb G1 and G1 SCTK from MACD with RBT phosphorylate. No division carried out without mitogen bound.
Phosphorylated RB will make sure transcription regulators of gene will be released and set the free. Here comes another cell proliferating from G1 to the S stage with mitogen around.
Hi everyone. So, what we can conclude on overall of this presentation? Here we go. Binding of RB protein with E2F as a complex, and this complex will binding with DNA sequence to prevent a transcription occur. Mitogen that as cell division will release a RB break and triggering activation of G1 CDKs and G1 SCDKs, but no division is carried out if mitogen is absent. RB protein also can involve in apoptosis. At the mechanism of RB protein in normal cells, RB is active at the G1 phase and inactive at S phase. Meanwhile, RB protein can be inactive by either mutation or hyperphosphorylation in a cancer cell. Retinoblastoma is the cancer that causes by mutation of RB protein. So, most mutation in the RB1 gene prevent it from making any functional protein. So, cells are unable to regulate cell division effectively. As a result, certain cells in the retina can divide uncontrollably to form a cancerous tumor. Just that from our group, and thank you so much.